So without further ado, here is our last lesson on techniques in research, learning actual practical lab skills that you'll all learn when you work in wet labs if you follow the steps from our last lesson. So again, I just wanted to say it has been a pleasure teaching you all. This is our last lesson in a partnership between Voices on the Mount and Junior Medical Academy. And all of you from across the world, from 12 countries, it's been a pleasure teaching you, and I can't wait to go and delve into our lesson right now. So today we're going to learn about three main topics. Firstly, transformation, which basically looks at how do we clone the DNA we have? How do we make more of a certain amount of DNA by putting it into bacteria and making the bacteria divide? That'll be interesting, by transforming it into bacteria. The second thing is called mini preps. So this has many names. It's either called mini preps, Midi preps, or Maxi Preps, depending on how much DNA you want. But essentially, all these preps basically do is it answers the question, how do we take DNA from a bacterial cell and then extract it from a bacterial cell so we just have DNA? So that's a very important, very fundamental procedure. It's actually the first lab process that I ever learned in my life when I worked at a wet lab. So it's very basic, very easy, but you just want to understand a little more conceptually how it works to understand the procedure. We'll be going over that. And lastly, we'll do Western blot. So for those of you that joined us in our last lesson on gel electrophoresis, Western blot is actually kind of similar, but it essentially asks the question, if I have a mixture of multiple proteins, like in your blood, you have hundreds, if not thousands of different proteins. If I have a mixture of tons of different proteins, how do I isolate a specific protein that I'm interested in and see if that's in the mixture? This can have many applications, ranging from, do I have a cancer protein in my blood? Do I have, HIV, AIDS, that kind of thing? Do I have, even the coronavirus and any proteins that produces? So this has a host of applications and very, very important tool. And we'll be saving that for last. So without further ado, we're going to start with transformation. And again, transformation at the higher level asks the question, how can we take any DNA we're interested in? And yes, this includes human DNA. And primarily, one would argue it's for human DNA, for using it for medicine. And how do we make more of it? So again, this answers the same question as PCR, so polymerase chain reaction. But a difference I want to note here is that while PCR, polymerase chain reaction, can make lots and lots of DNA, it is error prone. There is a percentage of the DNA that you're going to create that has some mutation that's inaccurate. But with transformation, it's highly, highly more accurate. So it's more likely, if you're using transformation, that you won't have mutations. It's safer, and it's better to do. So that's why this is more, if you want precisely the exact kind of DNA that you want and you are very keen on not having any errors whatsoever, transformation is probably the way to go. And most of you will probably do this when you work in a lab. So with that, before we delve into the specifics of it, we're going to view a case and see how we can solve this case through our lab work by delving into it. So we're going to start by covering Laura's story. Now, Laura, to give you a quick 1-minute overview, was she was a very kind, very, very peppy, very active young girl in elementary school. Laura loved to play. She loved the outdoors, and she just loved the sun. One day, while she was playing in the playground, she experienced some interesting symptoms that she never felt before, and that kind of worried her. She had some weakness and fatigue while she was playing hopscotch, and she's usually a very active girl, so this was new for her. In addition to that, she found herself getting her vision getting blurry and found herself getting dizzy. And again, she just started playing, so this didn't make sense to her. So as she was actually going to the bathroom to go and freshen up, she actually fainted, and that was really, really scary. So when Laura went to the hospital that day, and her parents drove her to the hospital, the doctor diagnosed her with type 1 diabetes. Now, diabetes is basically and fundamentally a mutation, is a error in the insulin protein. So basically, there are two proteins in the body. There's insulin and there's glucagon. So I shouldn't call them proteins, they're hormones, but like there are two, we'll call them substances in the body, insulin and glucagon. So it's like a seesaw almost. It's like a yin and yang reaction. What glucagon does is that glucagon helps to release more sugar in the blood. So when after I eat, let's say I eat a chocolate bar, glucagon helps to take the sugar from that chocolate and put it in my bloodstream. This is a good thing because we can't survive without sugar. But as with anything in nature, too much of one thing is a bad thing. So that's what insulin comes in. Insulin actually packages all the sugar in my blood and takes it away from my blood. so that it doesn't have to go through my circulation. So that's how there's like a back and forth reaction of glucagon releasing sugar into the blood, and insulin packaging that sugar in the blood, so that we have a perfect balance of sugar in our blood. But when you have type 1 diabetes, what essentially happens is that your insulin stops working. So you might have, glucagon release lots of sugar in your blood, but you don't have a molecule to store down all the sugar in your blood. So then that can get dangerous, because if you have too much sugar in your blood, that can lead to defects in the in vision, because too much sugar can affect the retina. That can also lead to hypertension and a lot of organ damage disorders. So that's very dangerous. So let's look at how we can use transformation to go and get more of this insulin gene. So essentially, our gene of interest is the insulin gene. So let's look at two things that transformation does. The first thing that transformation does is that it makes more DNA for us to analyze. The second thing it does is that it makes more protein for us to use for treatment if needed. So again, the reason why this happens, so at a higher level, all transformation is doing is I'm taking DNA that I'm interested in, I'm putting it inside of a bacteria, and then the bacteria is making clones of that DNA for me. So the reason why we also get more protein is this central dogma of biology that we've covered almost every other lesson. So just to give you a quick 30-second review, just to freshen our memories, DNA is really useful for division, right? It's very effective for cells to divide with, to divide and allocate properly. But it's not in a language that the cell can understand. So for many of you, let's say many of you don't speak Spanish. So let's say that this is in a language you don't understand. So that's like, imagine that you are like transcription factors in a cell. You're the cell. You can't read this. You can't make sense of this. That's why we have factors in the cell that convert DNA to RNA, which is a language we can understand, like in English. And these are instructions for making proteins. And in this analogy, the proteins can be thought of like cake. So if I follow these instructions of adding flour, adding eggs, adding sugar, then at the end, we'll get a nice whole cake. That's the end result that we want. So if I say that through transformation, if I'm making more DNA, then as a result, if I have more DNA, I'll have more RNA. If I have more RNA, I'll have more proteins. So that's how through transformation, I can get more DNA as well as more proteins. If my bacteria have more DNA, when they're reading and interpreting the DNA like a recipe, then of course they're going to make more proteins. Think of it like this. If I gave you more cake recipes, if I gave you 20 cake recipes, and let's say you were an amazing baker that never got tired, then you would make 20 cakes. If I gave you 100 cake recipes, you'd make 100 cakes. So that's how if I'm increasing the amount of DNA I have, I'm increasing the amount of protein I have. Pretty simple. Just wanted to make sure we're all conceptually clear on that. So now this is going to seem kind of random and stupid, but I swear this is very, very relevant, so just bear with me. So I'm going to tell you the story of a magic oven. So this magic oven is you came across it, some genie granted a wish, and no matter what happens, every few seconds, what happens to this oven and the pizza inside, what happens to the oven is that it doubles. So how many ever ovens we have, they always double in quantity. And if you notice, every time the oven doubles, it doubles the contents inside of it as well. So when we have more ovens, we also get more pizzas. So it sounds stupid, but I hope everyone can see what I mean by this, right? We have an oven with pizza inside. Then we have two ovens with twice as many pizzas, 4 ovens with 4 times as many pizzas, 8 ovens with 8 times as many pizzas. And then let's say you discover that, you know what, I hate pizza, but I actually really like roast chicken. So let's say you replace the contents of the oven. Well, no problem, because the oven constantly doubles and the contents inside of the oven also double. And you realize that, well, this is really cool. I can put anything in the oven and it can, double that for me. And it exponentially grows. It goes from 1 to 2 to 4 to 8 to 16, and it keeps growing like that. Well, maybe one would hope we would put like $100 bill in the oven and we'd get more money, right? By the logic of this kind of magic. So so if you haven't caught on already, this is very similar to the way that bacteria divide. So in this analogy, the oven is the bacteria, and the roast chicken or the pizza or whatever is inside of the oven is a bacterial DNA / plasmid. So in this analogy, this is actually very true to what happens. When we have bacteria and the DNA inside the bacteria, the genetic material, when the bacteria divides, we have the genetic material also divides. So as the bacteria keeps on dividing and dividing, we get more and more of the DNA. So then scientists realize that, hey, if these bacterial cells are just dividing randomly whatever genetic material we have inside of them, then why don't we put something interesting that we want more of? Like, who cares about bacterial DNA? Let's put more of insulin genes inside the bacteria and see if that works. So what we realize is that bacteria are what we like to call incompetent. And if you guys know, that word incompetent is basically synonymous with stupid. But incompetent in this kind of sphere means that they're so dumb that they'll take up whatever we give it, right? So this is random DNA of interest that I have outside. So this is the bacteria, and the blue thing is the bacterial DNA. The red thing is the insulin gene. So if I put a random, DNA strand outside of the bacteria basically eats it up. It takes it inside of itself, and then it integrates it into its genetic material. So this is really cool because, well, if I want any gene, let's say I want to make more of a cancer-saving gene. Let's say I want to make more of a insulin gene. All I have to do is mix it in with a solution with bacteria. The bacteria just absorb it and integrate it into its own genetic material. So this is what that kind of looks like. I have the human insulin gene outside. So this is the human DNA. And this is what scientists thought was interesting, because the way DNA is read as a code, it works across, it can work across species, across bacteria to humans, humans to bacteria. And that is really, really insane. So if I have this human DNA outside and I have this bacteria, right, and the bacteria has its own genetic material, then all the bacteria will do is it'll take that human DNA up and it will integrate that human DNA into its existing DNA. And does everyone see how that works, right? The human DNA was outside, but then it takes it in and integrates it into itself. And that is a very cool process. And then from there, this bacteria will divide and divide and divide, and then we'll have more and more and more copies of this human insulin gene. So we can basically amplify the amount of DNA we have exponentially. So the way that those magic ovens were growing and growing and growing, and we had more chicken or more pizza, these bacteria keep dividing and dividing, and then we have more of this human insulin gene, or any gene for that matter. So, and the last step basically of transformation. So far we have, the DNA that we have inside, bacteria's taken it up, and we have tons and tons of bacteria. But one thing that we do want to check is that we want to make sure that the bacteria actually absorb this DNA. So about, not 100% of the bacteria will absorb this DNA. So we might have, let's say, 80% of the bacteria absorb the human insulin gene, and they're the bacteria that we want. But 20% of the bacteria didn't absorb the insulin gene, just because of a random reason, just because of random chance. So that's bacteria that we don't want, right? That's useless DNA for us. So how do we make sure that the bacteria that's growing is only the bacteria that took up the insulin gene? Does everyone see what I mean there, right? We're going to have bacteria that absorbed the gene and integrated it, but we're also going to have bacteria that didn't absorb the gene, that, didn't do anything. So they're just normal bacteria that we don't want. So what scientists do is that they actually do encode something called an antibiotic resistance within the gene that we put into the bacteria. So this gene would be integrated into this insulin gene outside. So we would modify this and put, the insulin DNA, and at the end of it, put an antibiotic resistance DNA strand. So what happens is that when this integrates into the plasmid, when this goes into the bacterial plasmid, we can put this bacterial plasmid with antibiotics. And I'm sure many of you know this, but obviously with the COVID pandemic, a lot of us have been using hand sanitizers to wash our hands. They're antibiotics, antiviral. All they do is that they're septic. They're basically poisonous for bacteria. So they kill bacteria. But this antibiotic resistance gene is like a magical shield. So it prevents the bacteria from dying from antibiotics. It basically gives them like an imaginary shield. It prevents, basically the way it works is that antibiotics degrade core enzymes and core proteins in the bacteria, which basically kill it from the inside. But the antibiotic resistance gene prevents that degradation from happening. So that's why it's really cool, because the bacteria that we have that didn't take up the DNA, that didn't take up the insulin gene, wouldn't have the antibiotic resistance gene, because the insulin gene and the antibiotic resistance gene were all combined. So those bacteria will just die out when we put on antibiotics. But the bacteria with this insulin gene and the bacteria with the antibiotic resistance gene can take it up and they'll survive. So this might be a little confusing, and trust me, this is confusing for me as well. So I came up with a perfect analogy for you all. So essentially, let's say that we're in a battlefield, and this is, this is like hell. This is like a death kind of scene, and there's just gunfire going everywhere. So let's say that, our JMA lessons taught you all this magical technique to put a shield around yourself and protect yourself from gunfire and protect yourself from this war zone kind of place. So let's say that I had a student at JMA who never attended my lessons, who never, watched any of the YouTube videos, and never learned the shield technique. So if I put them in this war zone battlefield kind of area, then they're going to get shot by this gunfire and they're not going to make it out alive. They're going to die immediately because they didn't pay attention for the JMA lessons. And let's say concurrently, I had another student who actually attended JMA lessons, who was a good student and learned this shield technique. And then they were able to produce this shield and protect themselves from this deadly gunfire. And then they emerged out victorious and they came out alive. So if I didn't know, like I, from the instructor's point of view, didn't know who attended my lesson and who didn't attend my lesson, I could put both of these students in this battlefield kind of zone. And the students that died from this gunfire that couldn't do the shield technique, I would know, well, if they can't do the shield technique, then they never attended my lesson. So this student obviously never attended a JMA session. But the students that came out alive and that had the shields technique and that could live because they produced the shield, I could say with confidence that, well, this person, because they survived, definitely attended JMA lessons and they learned this shield technique. So that's a little gory. Obviously, I hope none of you ever, I wish death upon none of you. But I say this purely for the analogy that this is exactly what happens with bacteria. And this is what we're doing with transformation. So the bacteria that, in this analogy, attending JMA lessons means bacteria that took up the insulin gene plasmid and that took up the antibody gene resistance. And this is an analogy for bacteria that didn't take up the insulin gene. So, and this is obviously antibiotic, right? So if you did take up the insulin gene, you're protected because you also have the resistance gene. And if you didn't take up the insulin gene, then you don't have the shield technique, you don't have the resistance gene, and you'll die from antibiotics. So that's how we know that, that's how we know if a bacteria took up the insulin gene or not. And then at the end, the only bacteria we have left are only the survivors, right? Only the ones that survive the antibiotics. And those are the ones that we go with. So these are examples of antibiotics. I only included the names, not for you all to memorize, but just so you know the names and you're familiar with them. So ampicillin, kanamycin, tetracycline, and they're often abbreviated as AMP, A-M-P, CAN, K-A-N, T-E-T, TET. So they're like short form names on the tubes, just because we like shorter form names for simplicity. And the antibiotic resistance genes are pretty intuitively called ampicillin resistance gene, kanamycin resistance gene, tetracycline resistance gene. So pretty self-intuitively, if I have a bacterial plasmid that has the insulin gene and a ampicillin resistance gene, when I put it in ampicillin, which is a kind of antibiotic, that bacteria will survive because it has an ampicillin resistance gene. So that's how I know that it absorbed this plasmid. And that's how it works in a lab type setting. So that's what we have for transformation. So that went actually surprisingly, quickly, pretty smoothly. So I'm going to pause for just about 10 seconds here to let anyone ask questions. So you can type that into the chat. You can ask it out loud. We'll take a quick 10-second break before we go to the second procedure. Awesome. Yeah. I'll be moving on now, but feel free to continue typing your question if you're currently typing. So, and I hope this kind of makes it clear, elucidates for you all how this could help for Laura's story, who was a diabetic. So for her, let's say that we had a lack of insulin, right? We could go and clone this insulin gene and make multiple hundreds, if not thousands of copies of this insulin gene within bacteria. So then the question might be asked, how do we isolate this DNA from the insulin DNA, the insulin gene, from the bacteria? So that's the next step. And we answer that with the topic of Midi Preps. So Midi Preps, Mini Preps. So again, there are three names for this: Mini Preps, Midi Preps with a D, and Maxi Preps. So that basically means how much DNA do we want in the end. Mini Preps gets us a little bit of DNA. Midi Preps gets us a middle amount of DNA. And Maxi Preps, as the name suggests, Maxi, gets us a lot of DNA. So it depends on, do you only need a little bit, a medium amount, or a lot of DNA. But all the processes are basically the exact same. But they all ask the question, how do we harvest DNA from the bacteria? That, okay, great, we have, let me find an image of this. We have bacteria cells with, DNA inside of it, but bacteria have all this cytoplasm, all this proteins, all these transcription factors, all this RNA, all this organelles inside of it that we don't want, that we're not interested in. So we're asking the question, how do we isolate the DNA and only get the DNA and, basically wash away everything else? So that's what we answer with mini preps. So to answer this story, we're actually going to go and use another case study. So we're going to study Kyra's story. So Kyra, as you can all see here, she is a loving mother. Loving mother of 5. She has a beautiful family, has a very happy life, but she has something called the BRCA gene. It's pronounced BRCA, but it's BRCA. So this gene basically makes you very highly predisposed to having breast and ovarian cancer. So for the BRCA1 gene, there's 2 variants, BRCA1 and BRCA2. But for the BRCA1 gene, if you have this gene, you have an 85% chance of having breast cancer and a 25% chance of having ovarian cancer. So that's really, really insane. So over 8 in 10, over 4 in 5 women who have this gene will get breast cancer, and about 1 in 4 women who have this will get ovarian cancer. So it's a very, very dangerous gene. And obviously, basically there's a defect within the DNA. So let's say that you as a scientist were working in this big study project as to how can we fix this DNA for the BRCA gene mutation? How can we do gene therapy and fix this DNA? And your colleague comes and says, hey, I got the DNA fixed. I got the BRCA gene fixed, right? I fixed the BRCA gene. I have this DNA strand that has a fixed BRCA gene, so there's no risk of breast cancer, no risk of ovarian cancer, and I have millions and millions of bacterial cells that have the fixed DNA inside of them. Now go do your thing. So now you'll say that, well, my colleague obviously used transformation to get the DNA inside of this bacteria, but what do I do now? What do I do now to get this DNA outside of the bacteria? Because I only want the DNA to put into the patient. I don't want the bacterial cells to put into the patient. I don't want the bacterial proteins because that could harm them. I don't want the bacterial, RNA or the fluid or cytoplasm or whatever. I only want the DNA. So that's where mini preps come into play. So I'm going to repeat this a bunch of times and give you guys analogies. So not all of this is immediately going to make sense, but just bear with me. So essentially what happens is that first we have lots and lots of bacteria. So we spin everything down into a centrifuge. And what a centrifuge is essentially it's this kind of machine. So it looks like this. It has these kind of holes in here that we put the epitubes in. And we covered epitubes last time. So epitubes are basically just like really small cylinders that have like DNA inside of them or that have anything I'm interested inside of them. So we can put the bacteria, all our bacterial colonies and all that into these epitubes, and we can put them into this centrifuge. So what the centrifuge does is that it spins around a bunch of times, and it basically spins the heaviest contents to the bottom and has the lightest fluids towards the top. So think of it like this. If I had like water and honey in a jar, then if I actually no, this is a better example. If any of you have ever gotten, let's say, smoothies with toppings on it. So let's say I got, like smoothies with, I don't know, like blueberry toppings inside of the smoothie. Then if I spun everything down, then all the heavier blueberries will sink to the bottom. Or let's say I had water and I have like, strawberries floating on it. And if I spin everything down, then the heaviest things and the strawberries will come and settle to the bottom and the water will settle to the top. And here's an image of how this centrifuge stuff actually works. So essentially, and so
you
really
can separate components of a solution such as plasma and red blood cells from a sample of blood. In a lab, tubes of blood are loaded into a centrifuge in pairs. Samples are loaded in opposite positions to help balance the centrifuge. The spinning forces the heavier components, such as red blood cells, to separate from the plasma solution and collect at the bottom.
Oh, that interesting. I don't know why that weird image is there. I apologize for that. But yeah, and the lightest things basically rise to the top. So I see there might be some question in the chat, so I'll take them now. Oh, okay. Can everyone hear me right now? Okay, great. Sorry, I just realized the video may have been covering my voice, so I do apologize for that. I wasn't really saying anything of use that the video wasn't mentioning. But essentially all I was saying, and also I apologize for the weird image at the end. I wasn't aware that popped up. But essentially all I was saying was that, as you can all see here, like in these little tube holes, we put in the epitubes. We put in the tubes and then that have the bacteria in them. And then we basically spun it down. So we basically mix this concoction in a circle. And what happens is that as soon as you kind of mix this in a circle, and I'm sorry, this bottom part is kind of happening. Give me one quick second. Okay, there we go. Better. So essentially what we did was, just to reiterate, we put these epitubes that had, bacteria inside of them in this machine. It spun it around a bunch of times. And what happened as a result is that the contents, the heaviest contents, like the DNA, the proteins, sink to the bottom, and everything else that's lighter, like the cytoplasm, the extra fluid, the cell matrix, that all floats towards the top. So the steps here are as follows. We spin everything down in the microcentrifuge. Then we have, the heaviest things like the protein and the DNA at the bottom. Then we add a bunch of buffers to clean out the waste. So as I mentioned, we have proteins and DNA and a bunch of waste that we don't want at the bottom. So we can add a bunch of buffers to dissolve away like proteins, dissolve away, different organelles that we don't want. So we're only left with DNA. And then we can drain this waste through filters that, the proteins will pass through and the DNA will get caught in. And then in the filters, we are just left with DNA. And that's how we isolate DNA from the bacteria. So this is not going to make sense to a lot of you, or this might be confusing to a lot of you that's never done the process. So I'm going to explain this in multiple ways, primarily through this really dumb analogy. So initially, I'm going to give you, again, this is going to sound very stupid, but so someone asked, what are buffers? So we're going to explain this. I'm going to explain this in just one bit. So in a lot more detail. So let me explain the analogy first, and then this will start making a lot more sense. So let's say that I have a jar full of feathers, and inside of this jar, I have a marshmallow with a marble inside of it. And let's say this marble is my marble of interest, and I really want to isolate the marble. But let's say, everything is too small. I can't reach my hand inside and do anything. So I have to do this through like the way a scientist would do it. So firstly, that this jar is filled with feathers. Secondly, the marble is somewhere in between, like in the middle of the jar, and it's inside of a marshmallow, and that marshmallow is somewhere inside of the jar. So what are you going to do to make sure that we can get the marble? So firstly, what we're going to do is we're going to shake the jar. So once we shake the jar, what's going to happen? So the heaviest things, like the marshmallow with the marble at it, is going to come to the bottom. And then the lightest things, like the feathers, are going to get displaced towards the top. So now once I shake the jar, my marshmallow and marble is going to be at the bottom. My feathers are going to be at the top. So now what I can do is I can dump the feathers. I see all the feathers at the top. I can just pour it out, right? I can get rid of the feathers. And then I have my marshmallow with the marble inside at the bottom. So that's all great. Now I know where the marshmallow and the marble is, but here's the issue. I don't want the marshmallow. I only want the marble inside the marshmallow. So what I'm going to do is I'm going to put a bunch of chemical solutions to dissolve away the marshmallow. So, in this case, I chose Coke because Coke is very, it's acidic, it's fizzy, it's carbonated, so it can actually melt away the marshmallow. And if any of you have tried this experiment, if you put a marshmallow inside of something like Coke, Coke is actually really bad for your teeth. It damages your teeth. So if you put your tooth inside of Coke, it'll dissolve away completely. So if you put a marshmallow inside of Coke, it basically dissolves away the marshmallow over the course of time. So let's say I put all these chemical solutions like Coke and stuff into this jar, then it dissolves away the marshmallow, right? And then I'm left with this marble inside of this weird liquid solution. So then I can dump away this dissolved solution, this dissolved Coke and marshmallow solution through a strainer. So then all that's left once I dump that away is the marble. And then we've isolated the marble. So that might sound kind of, dumb and stupid and a little silly, but this is a perfect analogy, or a close to perfect analogy, for how we're harvesting DNA from bacteria. So I'm going to go over all of this and show you how this works with DNA. So in this analogy, the jar full of feathers is everything else. The jar is the cell. The feathers is everything inside of the cell, like the cytoplasm, the transcription factors, the organelles, everything else that we don't want. The marshmallow and the marble inside of it represent the casing of the DNA, and the marble is the actual DNA that we want. So again, the marshmallow and the feathers are basically all the extra stuff that we don't want, that the DNA is hidden in, and the marble is the actual DNA that we want. So when I say shake the jar and we get the marshmallow with the marble to the bottom, then that's basically the equivalent of centrifuging. So when you centrifuge, this exact same thing happens, where the heavy stuff like the proteins, the DNA settle to the bottom, and the light stuff like that we call the supernatant, the light stuff like the cytoplasm, the liquidy organelles and all that all settle to the top. So that you can literally just dump out, and you literally just dump it out like that in a pouring motion. And you're left with just like the heavy stuff, like the proteins and the DNA. So then what you do is you add a bunch of buffers, and buffers are like chemical solutions. They're chemical solutions that are meant to dissolve and react with certain kinds of molecules. So what those buffers do, obviously they're not Coke, but they're actually scientifically made buffers. that react with the proteins and the other material and dissolve them away. And they don't dissolve away the DNA. And what happens is that you can pass these through a strainer, you can pass all that through a strainer, and you're left only with the DNA. And that's how we isolate the DNA from the rest of the contents of the cell. And someone asks, I would like to ask what the behavior of the bacteria will be if the antibiotic DNA is integrated already into the existing DNA. That's a very, if the antibiotic DNA, oh, you mean the antibiotic resistance DNA? That's a very good question, Edwina. So essentially, this is going a little bit back. So essentially, and this is probably a good place to answer that question as well. But what happens in the bacteria, if we go back a little bit, so just to reiterate the question, Edwina basically asked, how does a bacteria behave differently if we have this antibiotic resistance gene inside of it? So the reason why bacteria die because of antibiotic solutions is that these antibiotic solutions, they don't just like explode the bacteria, although some do, but they basically inhibit the synthesis of certain kinds of key proteins inside of the bacteria that you can't survive without. So the equivalent of this is that in humans, let's say I gave you like cyanide or poison or something. Poison doesn't like make you explode, right? What poison or doesn't make you dissolve as a human being. What poison does is that it can stop your heart, or it can prevent your body from taking in oxygen. So you just don't get oxygen, your heart doesn't beat, you just die because you can't survive without that. That's similar to what it does to bacteria. It basically kills away key proteins inside of the bacteria, stops division, stops protein synthesis that the bacteria cannot survive without. And the bacteria just die with that. So what the antibiotic resistance gene is that it codes for counter proteins, and it stops the degradation from happening. It prevents the antibiotics from killing away key proteins inside of the bacteria. The equivalent of this is that, if we had a poison resistance gene in humans, then it would stop, it would prevent our heart from stopping to beat. It would prevent our body from not being able to take in oxygen. So then even if the poison tried to make us, die from asphyxiation or die from, a heart attack, then this magical poison resistance gene would prevent that from happening. But that's basically how that alters the bacterial function. That was a really good question. So back to isolating the DNA from the bacteria. And so the buffer is made specifically to dissolve the DNA, but everything else. Yes, exactly. So someone asked, is the buffer made specifically in this instance to dissolve everything but the DNA? And that is exactly right. So again, this marshmallow represents everything else, like the heavy proteins, the heavy organelles, the DNA, and this buffer basically dissolves away everything else, and you leave only the DNA. So it doesn't harm the DNA. So the way that this process works tangibly is that, well, we have our epitube, and the epitube has all the bacteria inside of it. So this is a bacteria with all the proteins, transcription factors, DNA, cytoplasm, all that. So then we basically spin it down. So this spinning motion basically means that we put in the centrifuge and we spin it down. And what that does is that it lyses or breaks open the bacteria, and it has a pellet and a supernatant. So basically, the pellet is the heaviest thing. This is like the marshmallow. This is the heaviest things that settles to the bottom, and the supernatants are like the feathers. These are the lighter things that float to the top. So the pellet is actually like hard. So it basically settles to the bottom and it sticks to the bottom. The supernatant is like a soup. It's literally liquid that's at the top. So, and this is genuinely what I did when I did this mini prep. I took this tube and I dumped out the rest. I dumped out the supernatant, just like liquid. And the liquid like soup just like flows right out of the epitube, and the pellet sticks to the bottom. So then you're left with only this pellet at the bottom. And this pellet is the heaviest material, like the DNA, like the proteins. So then what happens? So then we basically are able to harvest this, and we add a bunch of buffers. So these buffers do is that they lyse the proteins. They lyse all the other junk that we don't want, and then we can kind of isolate the DNA from there. And then as a result, we can pass all of this through a specific filter that binds only DNA. So what happens is that when you pass material through this filter, if it's a protein, if it's like other material like that we just lyse, it flows right through. But if you're DNA, then you basically get trapped into this filter. So then we can basically pump this filter, and we have only DNA trapped inside of this filter. So does that make sense? I'm going to review that one more time. So we have bacteria. We spin it down so that we isolate the heaviest parts of the bacteria, like the DNA and the proteins, and we dump off the rest. Then we basically add buffers that dissolve away the proteins and the other junk that we don't want, but that keep the DNA intact. And the DNA is the red stuff here, that keep the DNA intact. And then we pass it through a special filter. So because we dissolved away the proteins, it's like a liquid. So it passes right through the filter. But because so this is a strainer analogy. So it passes right through the strainer. But if I put the marble and like the Coke solution into a strainer, the Coke goes through the strainer, but the marble gets trapped. So likewise, if I put this DNA solution through this filter, then the lysed proteins flow through and the DNA gets trapped. And that's how we isolate the DNA. It's called elution, but that's how we get the DNA and isolate that DNA. So for our patient here, who was suffering from the BRCA gene mutation, if we had tons and tons of bacteria that had the bacterial BRCA, like that had the perfect gene that solved the breast cancer, that didn't have the breast cancer mutation, we would basically spin it down so that that gene, as well as random proteins, were at the bottom. Then we would add buffers that dissolved away the proteins and put this through a filter. And then at the end, we would have this perfect BRCA gene corrected DNA that's trapped in the filter, and we could isolate that, and then we can use that as we wish, and then give it to the patient. We can use it for scientific treatment. We could use it for testing, but that's basically how that process works. It's a very, very cool process, and I will pause for about 10 seconds to see if anyone has any questions. And feel free to ask just about anything, either on the topic we just covered or on this topic. Okay, looks like everyone's following along now. Good, just continue typing into the chat if you would like. And let's go to our last thing for the day. So Western blotting. So this is basically answering the question, Western blotting. How do we detect proteins inside of a mixture? So let's say that we have a big mixture that's a combination of a bunch of different proteins, like your blood or your saliva. That has tons of different proteins in it. Let's say I want to detect one certain protein. I want to know if one certain protein is inside of your blood or saliva or whatever. And this can be used for cancer detection. This can be used for detecting HIV and that kind of stuff. And we do this process called Western blotting. And a lot of you will see some parallels between this and gel electrophoresis, but we'll get into that in just a bit. So we're going to start with Max's story. So Max is actually a college student. So Max is really living the college life. He's just about to graduate and receive his PhD in molecular biology. So he's been having a great time, and he also has a girlfriend. So Max just recently found out that he's been having some symptoms that might be indicative of HIV. And he's worried that this might progress, this might affect his relationship. And he doesn't, and because HIV is a sexually transmitted virus, a sexually transmitted infection, he wants to know because he doesn't want to affect his girlfriend adversely. So Max is essentially going to go to the hospital and ask them, well, I don't know if I have HIV. I have some symptoms that might be consistent with the disease, and I want to know for sure, do I have HIV? So then the hospital is going to tell him, the doctor is going to tell him, well, here, we're going to do a quick procedure to see, do you have HIV inside of your blood? So just as a note, there are many other procedures we can use for this. So the one that's used most commonly in hospitals is called CART therapy. CART for detection, C-A-R-T. But this also, you can also use Western blotting. It's a little longer, but I'm just using this as an example because this is a very important tool that we use in wet lab. So I'm just using this as a vehicle to explain that. So for Max's story, basically the top down of what we can do is that we can take some of Max's blood, the suspected patient blood sample. We can go and extract proteins from it, and we can take, we can spin it down and get only proteins that we want. We can load the proteins into wells, like inside of a gel, and we can basically go and try to see, is there an HIV protein within this blood sample? So I'm going to show you guys this graphic at least 2 or 3 times, explain it in depth. So if it doesn't make sense immediately, that is perfectly okay. But let's kind of start with what we have. So essentially what I'm doing is that I'm taking my blood sample and my protein samples, and I'm putting a quick dye in them, and I'm putting them into a gel. So I'm separating them by gel electrophoresis. So the use of this is that inside of this blood sample, I can't tell you where the proteins are. It's all mixed into this homogeneous mixture. So if I put it into gel electrophoresis, if I put it into a gel and apply an electrical current, then that stratifies the proteins. So then I can at least see in the gel where all the different proteins are. So this might mean that, I might have, an HIV protein. I might not have an HIV protein. So it's mixed, right? But at least I've separated all these proteins into this gel. The second step is basically transferring the proteins from this gel to something that has more of a solid support. So after we run this gel through an electrical current, it gets kind of weak and flimsy, and that it can tear really easily, and we don't want that. So we basically transfer, we press that gel into a filter paper, and a paper is more sturdier because this gel rips easily and this doesn't, so it has a sturdy back. And then this exact kind of protein signatures and all that are basically copied face to face on this filter paper. If any of you want an analogy, it's like if I was drawing, okay, this is a good analogy. It's like if I was drawing, it's like this. If some ladies might identify this analogy, but it's like if you put on a lot of lipstick and let's say you kiss your hand or something, then you'll see a mark of that lipstick on your hand. So you can still see all the lipstick that's on your hand. That's kind of similar to this, where we have all the proteins on this gel, and then that's kind of put onto this filter paper. So just the same way that if you kiss your hand, that lipstick leaves an imprint on your hand, that gel will, these proteins on that gel will leave an imprint on this filter paper. So that's this transferring process. And the third and last process is that we basically put on antibodies to detect the protein of interest. So we covered this in our very first lesson, actually, but antibody with oncolytic virotherapy. But antibodies are cool in that they only bind to a specific protein, and only that protein, and that one protein only. So we have, millions of proteins, and for every protein there is an antibody. So what we do is, well, we have, hundreds of proteins here. So in my blood, I might have proteins for proteins from the food I ate. I might have proteins that occur naturally. I might have proteins from my hemoglobin. So there's tons and tons of different kinds of proteins in there. And I might also have HIV proteins. So this might be Max's blood. One of these proteins might be an HIV protein. So if I use an HIV antibody that bound to the HIV protein, then what would happen is that this HIV, if I have the HIV protein, then this antibody will bind to that protein, and I'll know, well, did my antibody bind to anything? If it did bind to anything, I know that there's an HIV protein. If it didn't bind to anything, I know there's not an HIV protein. So these things are very, very selective, and they will only bind to one specific thing. So that's how we set them to only bind to the HIV protein. So some of you might be wondering, well, why are there 2 antibodies? So the reason for that is that this second antibody basically is selective for this antibody. So some of you might not have known this, but you can have antibodies that only bind to antibodies. So what happens here is, and I'll cover this in depth, so this might seem confusing, and this seemed really confusing to me the first time around. But basically, all that there is there's a protein. There's a primary antibody that only binds to a protein. Then there's a secondary antibody that only binds to the primary antibody, like the blue antibody that only binds to the red antibody. And once it binds, some of you might be able to see this yellow marker here. Once it binds, it initiates a color change reaction. So this is called horseradish peroxidase. It's a molecule that it changes color. It has a change in wavelength of light, which we register as a change in color whenever it binds to something. So that's how we know that, well, if there's a change in color, then that means that there's a protein for the antibodies to bind to. And if there was a protein for the antibodies to bind to, that means that protein was there. This is very similar to the biomarkers that we covered earlier, right? That if we see a change in color, then that might mean that you have the cancer biomarker. Or this is like the pregnancy test, where if you see a change in color on that line, then that means that, well, the antibodies were activated because you bound to a pregnancy hormone, HCG. So let's first start with an analogy, because you all know how much I love my silly analogies. So in this analogy, let's say that I snuck out of the house and I went to the beach. And let's say that my parents want to know, well, did I sneak out of the house and go to the beach? Because it isn't readily apparent by talking to me. So what they can do to see, well, did I sneak out of the house and go to the beach, is that they can analyze my shoes. So if I went to the beach, then theoretically I was on the sand and my shoes were on the sand. So what they can do is, well, if it's too close, it's too tiny to see. But let's say that if I did go to the beach, then I would have tiny, tiny particles of sand at the bottom of my shoes. So these we couldn't see with the naked eye because these particles of sand would be so small. But hypothetically, if I did go to the beach, then I would have tiny, tiny particles of sand in my shoe. So what could my parents do if they were being, thinking through like scientists? They could do is that they could first, because they don't want to ruin my shoes, they could have me step into dirt. So they can have me step and leave my shoe imprint into this kind of dirt mud background. So then what I have is this shoe imprint on this dirt mud background. And if I went to the beach, then I'll have these kind of sand particles that were these sand particles that are in this that are in the mud as well. So we have dirt and then we have sand particles. So then what they could do is that they could put in antibodies that are specific to only sand. And this doesn't exist, but let's pretend it does. Let's say we had magic antibodies that only bound to sand. And once they bound to sand, this antibody has a conjugate molecule, like this conjugate molecule, that if it binds to anything, it changes color. So if they basically put this solution all over this dirt foot imprint, and if there is dirt, then this antibody won't bind to it. So there's no color change reaction. But if I did go to the beach and there are sand particles, then the antibodies will change colors and you'll see specks of green inside of this dirt, which shouldn't be there. So then we can say that, oh, the specks of green are obviously specks of sand, which means that you did go to the beach. So what they can do at the end, that once we have, the shoe print, once we have identified the sand molecules, we can wash away all the dirt, and then we can see, that here is the sand that we have remaining present. And we can look at that sand and we can image that, and they can, frame it and make me feel terrible for the rest of my life and, ground me forever for going to the beach without permission. So that, that's very silly, very stupid analogy, but I think it serves to illustrate this purpose of Western blotting and finding proteins very well. So this step of leaving a shoe imprint in dirt is basically the equivalent of transferring the proteins from a gel to a paper backing. And obviously the proteins being in the gel, separating the gel, is like the sand being stuck in my shoe. And then the detection process of having antibodies being bound to my proteins is similar to having the whole antibodies being bound to the sand molecules and changing color. And through the color change reaction, we can see, well, are there proteins? The same way we were able to see, well, was there sand particles in the dirt? And obviously the way that we washed away this dirt and we just saw, is there any sand present, is similar to how we can isolate the protein from this gel kind of back. So this is like a overview, very not in-depth, like a very generic overview. So if you're like a beginner, then that's probably the extent of what you need to know. But I'm going to go a little bit more in-depth, both for beginner and advanced, because I think it's important just to go a little bit more in-depth, and I don't think it's going to be that confusing at all. So again, let's revisit step one. So again, remember that I had a blood sample from Max's story, right? Who's worried if he has HIV. So I have a blood sample that I'm basically taking the proteins from. So I'm spinning it down, I'm isolating the proteins, and I'm putting a dye into the proteins. I put the proteins into a gel. I put an electrical shock through the gel, and what happens is that all these charged molecules and proteins separate into this gel. So the last time we covered this with gel electrophoresis and DNA, but what we can do is we can isolate just the proteins by spinning it down and adding a different buffer that dissolves away the DNA and keeps the proteins. So then these proteins will all separate onto my gel. So that's my first step. My second step is that transferring these proteins from filter paper onto a solid backing. So all that happens is that I press this solid paper next to this gel and I leave it like that for like 60 minutes. So what happens is that these proteins, these protein dyes that were on the gel, transfer onto this solid colored paper, this solid filtered paper that has a thicker backing that I want to use. So that brings us to our third step. So I didn't cover this in the analogy, but essentially this is called blocking. So what I'm doing here is that I'm putting in, these blocking agents, you can use things called BSA, you can use literally milk. So all this does is that it clogs the pores inside of the gel. So to clarify why we're doing this, when we put on antibodies, we want them to only bind to the antigen of interest, the protein of interest. So the protein is the antigen. The antigen is another word for the thing the antibody binds to. In this case, it's the HIV protein. So we don't want our antibodies to accidentally go and land in the pores in the hole, in the pores of the gel, in the pores of the filter paper, or randomly, get stuck or bind to random things that we don't want them to. So that's why when we put on this milk, it's literally just milk, or we can use BSA or like other kinds of agents. What they do is that they clog the pores. So they make it so that it's even harder for the antibodies to bind somewhere random, fall into the pores, or to not bind to the protein. So obviously this milk and stuff won't bind to the antigen. It'll float through it'll fly right off of the proteins. So these proteins will be wide and exposed. And the rest of the material, the rest of the gel, the rest of the, filter paper will be blocked by this milk, will be blocked by these random molecule proteins like BSA or milk or whatever, so that the only exposed part is this antigen protein. So it really maximizes the chances of our antibody actually binding to our protein of interest. And it prevents these antibodies from sticking to the membrane or something and really definitely binding to our protein of interest. So once we do that, we have our target protein, we have our HIV protein. So then we add antibodies that we're interested in. So we add the antibodies, the primary antibodies we're interested in, and they bind to this protein exactly. What happens next? So these antibodies have bound to our HIV protein. The next, a secondary antibody that only binds to primary antibodies are added. So these blue antibodies will only bind to antibodies. These red antibodies will only bind to proteins. So we have this weird kind of sandwich up on top kind of structure where we have this antibody that binds to the protein, and we have the secondary antibody that changes color when it binds to an antibody. So the purpose of this antibody is to change color. The purpose of this antibody is to locate the protein. So you can think of it like, this is like a lost kid in a mall. This antibody is like the police officer that finds, the random parent that finds this lost kid in a mall. And they find this lost kid and they report it to the mall. And then the mall issues on an announcement and says, attention everyone, we have a lost kid at the mall. If you've lost your kid, please come here. And they announce it out loud. So that announcement is like the color change reaction. That's essentially how that process works. And once we have that substrate, we want that's the color change reaction. So this color change reaction is like fluorescence. So the way this works in imaging, and this is actually a real image of what this looks like in Western blotting, you can see that if it fluoresces green, for example, then you can see, well, here is the HIV antibody and the HIV protein. So if there's no green on here, then we know that we're safe. Max is safe. He doesn't have HIV. If it is green, we know that Max does have HIV. And that's how we can kind of diagnose him. And this is also just another diagram illustrating that point. We have this mixture. We separate the proteins by electrophoresis into the gel. We transfer the proteins from the gel to a membrane. And this is a real life image of what that looks like. We have the paper, we have the gel, we kind of stick them next to each other. And we add these antibodies that the first one to bind to the protein, the next one to glow out loud, to glow through like a green color or something, that we can detect what kind of, is that protein present. And this can also be used for multiple purposes like oncolytic virotherapy. I'm not going to go too in depth here, but we can use this to detect certain kinds of proteins that we want on top of our virus cells, or certain kinds of presence of cancer proteins through Western blotting. And again, we don't need to go too in detail there. That's not too relevant. But that's essentially all we have for today. So again, just to kind of recap, the three things we covered was, firstly, we covered transformation, which answered the question, how do we get DNA, put it into a bacteria cell, and get more of that DNA? So if we have like an insulin gene or a cancer-solving gene or something, we can, like a treated version, like a repaired version of the BRCA gene, and we can have lots and lots of that gene. So we can use this for two purposes. Either we can use that DNA to analyze, to study, to do more research on, or we can use that DNA to make proteins. And we can use, let's say, make the insulin protein and give it to Laura to solve her, to help her with her diabetes. The second thing we covered was, the process of mini preps. So how do we isolate DNA from bacteria? So if I give you lots of bacteria with a BRCA gene, the repaired BRCA gene, how do we harvest the DNA from that bacteria and just have the DNA? Wash away the proteins, wash away the transcription factors, pour away the cytosol, and be left with only the DNA of interest so that we can use it again for experimentation or for gene therapy for patients with the BRCA gene. And lastly, what we just covered is Western blotting. So how can we see if a certain protein is present in a mixture? Just by looking at your saliva. Can I tell you, do you have lung cancer? Just by looking at your blood, can I tell you, do you have, pancreatic cancer? Do you have leukemia? Do you have HIV? So all that process does, what Western blotting, what we just covered does, is that we take our proteins, we take our mixture of blood or saliva, we isolate the proteins, we put it through a gel so that all the proteins are separated physically. So we have some proteins on the bands over here, we have others here, we have others here. And then based on that, can we basically put on antibodies that change color when they bond to a specific protein? So if my blood doesn't have HIV, the antibodies aren't binding to anything. So I don't see a color change reaction. I don't see any colors. So I can say, hey, I see no colors, you don't have HIV. Or if I do see a color change reaction, I can say, hey, I see a color change reaction, therefore you have HIV. So that's what we have for today. That's all for today. And as a lasting note, this was our last lesson, but I just wanted to say it genuinely and truly has been such an honor and such a privilege to be teaching each and every single one of you. I just can't believe how much all of you have grown throughout the short time, the few months that I've gotten to know you all. But just from seeing the level of questions each of you have asked from, starting from, well, how does DNA work, all the way up to, well, how can we use organ therapy to go and help cure people with serious cancer disorders? How can we detect cancer and help, with gene therapy processes for people with breast cancer mutations? It's really, really been an incredible, incredible process. And really, I'll be sending out an email soon with a post test for you all to do if you're interested to see how much you've learned. I remember the pre-test a lot of you struggled with, and they seem like really hard questions. So I'm going to give that again to you so you can all see, well, how much have you learned, how much have you grown, and how much do you now know as a result of these classes. So just two really quick things that I did want to address. I know some of you are asking questions in the chat. I'll get to those right at the end. First is the mentorship program. So for the mentorship program, I know I asked some of you, I posed this in the middle of the lessons, but I said, if you're only interested in SAT or ACT help, you can sign up for the mentorship program. So we actually renovated our website. We kind of solidified our mentorship program, but I'll send it a link to all of you soon. It's at jma.education/mentorship program, /mentorship, sorry. And I'll send it out to all of you, but I want all of you to sign up for the mentorship program if you're interested. So I'll pair you with a university college student to give you any help with the ACT or SAT. So for any kind of standardized testing, for applications to scholarships, and we'll provide you with a list of potential scholarships you can apply for. And they'll help you with learning more about any kind of academics, like learning more about STEM fields. So they'll basically go and help teach you more about medical biotechnology and give you more elaboration on the material we covered here. And they'll even help you with college application material. So they'll help you navigate the common application process, help you apply to universities, help you with essays that you're interested in. And even if you're in, even if you just started secondary school, you just started high school, they'll help you with how to approach high school, how to, find what you're passionate about, and have a good profile to go and apply to US universities and Ivy League universities. And I'll send out that link. So that might be interesting to all of you. And I'll kind of show you all here. It's jma.education/mentorship. So I'll send out this link. So no need to copy it down or write it down. But essentially, this is our mentorship program. It's our website. So over here, you can sign up as a mentee. So mentee is you who will get the mentorship. And the mentor is the mentors that we've already had sign up that are interested. So we'll give you help with academic and STEM mentorship, standardized test preparation, colleges and scholarships. And, this is just more information about how, this will all be available to you. We'll do all the busy work. We'll handle the logistics. We'll go and help you connect with your mentor. We've already been operating for tons of people throughout 5 countries. And there's a quick FAQ stuff. So how you meet your mentor, is it free? And we'll answer all these questions, and I'll send this link out to you. So that's a question of the mentorship program. And again, I highly recommend all of you sign up for this program because it's really helpful, really cool, and we give it to you for free. So we'll never charge you anything ever. And we really just want you all to learn and grow and have the resources that, students here have, and students at private schools that, pay hundreds of thousands of dollars are able to have. And the second thing I wanted to address was the JMA Ambassadorship Award. So through all all of you that regularly attend the lessons, that are keeping up, that ask questions. I can't tell you how much I appreciate it all. It's really, truly such an honor. So I will be reaching out to those of you that have got ambassadorship privileges. So I'll send you a link with all the information you need to know, all the steps to proceed, a Google form to sign up and confirm your ambassadorship, and what the ambassadorship means. So I'll give you a quick 30-second overview. The ambassadorship basically means that you've been chosen by us to be a local chapter president, to be a JMA leader in your community. All of you, because you've taken this interest to actually go and learn this material outside of school on your own terms and be experts in biology, in medical biology, and do some tangible research in the world, you're uniquely situated in your community to go and spread this knowledge out to other people in your community. So what this will entail, and we'll lay this all out for you, we'll explain and help you through every single step. But what this will mean is that we will give you all the material, all our PowerPoints, all our teaching guides, all our lessons, and the YouTube links of how to teach our lessons. And we'll help you become a local chapter leader so that you can get everyone in your community involved in things like our mentorship program. Everyone in your community to learn more about stem cells and organ research and these wet lab techniques, and learn about how to reach out to professors to get them involved in science fairs. And we'll give you actually a science fair opportunity and to actually spectate one, to get access to scholarship opportunities, and really create a brighter future for the students in your community. So we recognize that a lot of you are from really beautiful places in the world that we as JMA aren't currently operating in, like to name a couple examples, there are some students here from Botswana, there's some here from the Gambia, there's some here from Namibia, there's some here from Nigeria, from Kenya. So we don't currently have branches that physically operate in these locations, but we would love to work with you to help create this educational access for free to these students. And as students who have taken this initiative, we think you are uniquely situated to be the leaders in your community and actually enact this kind of change. So that's why we want you to be these JMA ambassadorship, to hold these JMA ambassadorship privileges. So again, I'll email this out to you. So everyone, please keep this in mind for email. Please keep an eye out. So we'll send this out soon. But that I really wanted to mention and stress that we'll be sending that out to you all, and it'll definitely be worth your while. So again, I just wanted to end on that note that thank you so much for all your help, all your participation, all your devotion. Really, it's rare to find this kind of love and passion of science. But the last thing I did want to mention is that definitely sign up for the mentorship program. But even if you don't, or even if you do sign up for the mentorship program, I'm always open personally to each and every single one of you to help you get research internships, to help you all get paired with professors, and to help you all actually get tangible college research at universities, at top-tier Ivy League universities. Not only to help your profile and help you get scholarships and get into universities like Ivy League University when you apply, but also to solidify your knowledge of science and help you contribute something tangible. It's one thing to learn something on paper, but it's another thing to apply it and do actual work with it to see how you like it. Because we want to raise the next generation of scientists and next generation of researchers. So what I'll do personally is I'm actually currently reaching out to some university professors at Brown, Harvard, and MIT to see if they'll be receptive and open to taking on some of you as interns. So what I'll do is I'll let any of you know if I have opportunities from professors that take any of you on as interns, and I'll have you all sign up for these kind of internship spots and be able to do actual research with them. So because of the COVID pandemic, this will probably be remote research. So you'll be more like analyzing genes, helping to provide the theoretical basis, and doing computational biology work to support their research. But really, it's amazing work, very cool research. And I think all of you will enjoy it very thoroughly. So reach out to you with that. But in the meantime, please use our last lesson. That I would say that's probably one of our most important lessons on reaching out for internships, on reaching out to professors, and use that as the way to reach out to professors and write the email drafts and look at fields that you're interested in, and reach out to every university you can. So I gave you the examples of Stanford, Harvard, and Brown for places you can look at research, but you can look at any university. So you can look at any Ivy League, you can look at any state university, you can look at any private university. And if you look up a list of American universities, you'll see literally hundreds of universities. So genuinely, there's thousands of professors in the entirety of the US you can reach out to, each of which are doing their own unique, cool research. So please reach out to them, and I will help you all write your emails. You can reach out to me with any help, any time of the day, and I'll respond. I'll help you form your emails. I'll help you make your resume, anything you want. And I'll pair you with mentors to help you do that as well. So again, just thank you all for your time. And I think that's the bulk of it.